Okay, so let's get started. Welcome everyone to today's GlamCon seminar titled Sparsentan in IgA Nephropathy, an update on the clinical trials. We are thrilled to have Dr. Bruce Hendry speak on this topic with us. Dr. Bruce Hendry is a nephrologist and clinical scientist. He works for Travere Therapeutics as lead for nephrology in Europe. He is Emeritus Professor of Medicine at King's College in London. We are also thrilled and privileged to have two distinct panelists for this session today with us, Dr. Kirk Campbell and Dr. Shikha Wadhwani. Dr. Kirk Campbell is Professor of Medicine in the Division of Nephrology, Professor of Pharmacological Sciences at the Icahn School of Medicine at Mount Sinai. Dr. Campbell is the inaugural director of the Mount Sinai Center for Kidney Disease Innovation. In addition to caring for patients with kidney disease, he leads a multidisciplinary research program focused on understanding mechanism of kidney disease progression and clinical trials in the rare kidney disease space. Dr. Shikha Wadhwani is an assistant professor of medicine at the Division of Nephrology and Hypertension at Northwestern University Feinberg School of Medicine and is the founder of the Glomerular Disease Program at Northwestern University in Chicago. Dr. Wadhwani is the site PI for a number of multi-center clinical trials, as well as for the NIH-funded CURE-GN study. So thank you very much, and the floor is yours, Bruce.
Thank you very much indeed, Ali. I shall start to screen share. And I will try to remove the additional panels. So we have, I hope in a moment.
Perfect.
Okay, very good. Thank you very much, Ali, and thank you to GlamCon for inviting me to talk today on behalf of Travere Therapeutics. And also thanks to Shikha and Kirk for agreeing to act as expert discussants in this session. I recognize that this is a global event. And in the US, this is a holiday weekend, so particular thanks to the US participants for their enthusiasm. And I know it's also early on the West Coast. The agenda today is for me to talk about the Sparsentan program in IgA nephropathy. A quick introduction. Travere is a late-stage biopharmaceutical company looking into the development of treatments for rare disease. We're committed to providing better treatment for patients in these very difficult situations. And today I'm going to focus on one of our really major projects, which is the development of Sparsentan as a therapeutic, as a potential therapeutic in IgA nephropathy. Now, as we speak, Sparsentan is licensed in the United States by the FDA under accelerated approval for treatment of IgA patients at risk of rapid progression. In the rest of the world, it is not licensed. And recognizing GlamCon is a global organization, I'm going to be speaking about this agent in terms of the clinical data and clinical trials to provide you with evidence about the agent. And I'm not speaking about any individual or direct practical issues in the United States. This is supposed to be a talk where I'm going to give you the evidence base that we're emerging with Sparsentan to justify its potential use. We're very enthusiastic about the program. We think there's a lot of data emerging and continues to emerge. And so my objective here is to really convey that to you and to show you the evidence on which my enthusiasm is based. So first of all, some introduction to Sparsentan itself. Sparsentan is a new and novel mode of action agent. It is a dual endothelin angiotensin receptor antagonist. It is capable of binding to both of these receptors with high affinity and high selectivity as compared to ETB and AT2 receptors. And it is able to act at nanomolar concentrations in the aqueous phase, providing substantial antagonism to each of these two receptors and their downstream pathways. Why are we testing Sparsentan in IgA nephropathy? Well, there is a wealth of cellular preclinical molecular data showing that the pathways of angiotensin 1 and ETA are active in IgA nephropathy. These pathways are reinforcing each other with positive crosstalk, and the downstream effects of activation of these pathways include podocyte damage, endothelial glycocalyx loss, mesangial proliferation, inflammation, and fibrosis. So a range of reasons why these pathways should be considered targets in IgA nephropathy, as all of those 5 events I just mentioned are known to occur in IgA disease, and they do so from the time that the galactose deficient IgA is first deposited. I'm going to allow myself one preclinical slide to give you a sense of the range of data that we have from the cellular organoid and preclinical sphere, looking at sparsentan. This is a study in the GDDY mouse model of IgA nephropathy. These mice have been treated with either sparsentan in the middle column here, or losartan in the right column, at doses which create identical blood pressures. And at 16 weeks of treatment, 20-week sacrifice, the histology of the glomeruli showed clearly that sparsentan is reduced glomerular sclerosis, has protected podocyte number, and has protected the integrity of the endothelial glycocalyx. And on the right-hand side, you can see the statistics of these with the additional information that the sparsentan-treated mice have appearances which are now very similar to controlled non-diseased mice. This is just one example of the evidence that we have with collaborators generated that puts us in a very optimistic position to feel it is worth studying sparsentan in IgA nephropathy. So I'm going to talk about 3 main studies in this talk. The PROTECT study, the Spartan study, and studies of sparsentan and SGLT2 inhibitor combinations. Let me start off with the PROTECT study. The 2-year data from PROTECT was published in November of last year. You can see the title and authors here. And this reminds me to say that pretty much all the data I'm going to show you today is in the public domain, either in the full paper like this, or as presented at the recent ASN meeting in Philadelphia. There is a wealth of registry evidence that IgA nephropathy is a severe disease. These are data from the UK RaDaR registry. See on the left, the median kidney survival of IgA patients is about 11 years. And you can see on the right that a failure to control proteinuria in IgA patients leads to extremely poor outcomes, as shown in the green and amber lines, where proteinuria is above 0.9 gram per gram. Those UK data are fully supported by emerging data from the United States. As an example, this was work which was presented by Kathleen Tuttle's group at the ASN. On the right-hand side, you can see that IgA patients who have proteinuria above 0.7 gram per gram have a median survival before a major adverse kidney event of less than 4 years. So the RaDaR data and the US data emerging are very clear that IgA patients are in need of better than current treatment and standard of care, and that a focus on reducing proteinuria is likely to be a good avenue to improve their outcomes. So the PROTECT study was designed to test efficacy and safety of sparsentan using an active control design. The rationale I have already really covered, but I repeat it there, that targeting both the endothelin A receptor pathways and the AT1 angiotensin receptor pathways has a wealth of logic behind it in the context of improving and preserving kidney function in IgA disease. The study was a double-blind study, randomized, Importantly, with an active control arm of irbesartan, which was running parallel to the sparsentan arm. Patients to enter the study had to have biopsy-proven IgA nephropathy. They were adults, greater than or equal to 18 years, urine protein excretion of more than 1 gram per day, therefore high risk of progression, and an eGFR above 30. The study design involved a switch from their current standard of care to sparsentan in a single day. Importantly, patients to be recruited to the study have to be on a maximum tolerated dose of ACE inhibitor or ARB, and that this had to be at least 50% of the maximum approved dose in their jurisdiction. Following randomization, and the study starts with sparsentan or irbesartan, the two drugs were both titrated up the 2-week period at 200 mg of sparsentan, if tolerated up to 400 at week 2, and irbesartan in parallel, 150 mg per day, titrated up to 300 if tolerated at week 2. The study ran in a double-blind treatment fashion for 110 weeks, and at that point, study drug is withdrawn for a period of resumption of standard of care. And beyond week 114, patients could join an open-label extension, all offered sparsentan. The primary efficacy endpoint was the UPCR change from baseline to week 36 in the two arms. And key secondary efficacy endpoints were eGFR slope, either chronic slope, which is the slope from weeks 6 to 110, not including any acute effects in the first 6 weeks, and a total slope, which includes the first 6 weeks, day 1 to week 110. The patient disposition is shown here. You'll see that the randomization resulted in 202 patients in each of the groups. And during the period of the study, discontinuations are shown right and left. There were 48 in the irbesartan arm, 28 fewer in the sparsentan arm. A very substantial majority of patients completed treatment, 86% in the sparsentan arm, and almost all patients completed observations through the double-blind period, whether or not they'd had to stop treatment, over 98% in the sparsentan arm. So we believe this study was robustly conducted and that it provides a significant and robust test of the clinical properties of sparsentan in this IgA population. Here are some very simple examples of the baseline demographics. These patients had a median age mean age of diagnosis of about 40 and a mean age of initiation of the study of about 46. They were predominantly male. Systolic and diastolic blood pressures were moderately well controlled. They had substantial, 2/3 of them were on maximum labeled ACE inhibitor or ARB at screening. And this is a number higher than in most studies in IgA in recent times. Mean eGFR at baseline was about 57 ml per minute per 1.73 meters squared. And the urine protein excretion median was 1.8 grams per day. About half the patients had hematuria detected at baseline. This was a prevalent population. The study drug dose was titrated to a maximum, that is the 2 to 4 week period went up to the full 400 or 200 in over 95% of patients, as you can see on the bottom line here. This is the proteinuria data over the 110-week period. The interim result, the primary endpoint, was assessed at 36 weeks, is written in the middle of this graph, and the results were clear superiority of sparsentan, where about 50% reduction from baseline was seen, compared to 15% with irbesartan, a geometric mean reduction of 41% or 0.59. To the right, you see that the effect of sparsentan on proteinuria was sustained, and at the end of the study, the difference between sparsentan and irbesartan was fully maintained. Patients on sparsentan had a significant chance of reaching a complete remission event for proteinuria, less than 0.3 gram per day, as shown in the right-hand bar chart, 3 times greater chance at 31% than patients who were in the irbesartan arm. So significant proteinuria reduction was sustained over 110 weeks, and there was a significant chance in the sparsentan arm of achieving a complete remission event. Here is the data on kidney function. This is the eGFR data over 110 weeks by visit. And you can see that these are the mean change from baseline with standard error bars shown. And you can see that the sparsentan arm, which is in the green color, all visits, the eGFR is higher, and the difference from irbesartan appears to widen with time, showing an accrual of benefit. On the right, the bar chart shows the change from baseline to week 110. The sparsentan arm has a higher result. The difference between the two arms is 3.7 ml per minute per meter squared. The difference at 58 weeks was 1.7. And so these results show an accumulation of benefit with the improvement over the second year greater than that over the first year. These are the key secondary endpoints: chronic slope and total slope. Chronic slope, irbesartan 3.8, sparsentan 2.7, both negative. The difference favors sparsentan at 1.1. The total slope, the equivalent numbers, minus 3.9, minus 2.9. The difference favors sparsentan at 1.0. P-values there are 0.037, 0.058 respectively. So this slide shows you a totality of data that we have on renal function over a 2-year on-treatment period. Quick word about the p-values. One p-value is just above 0.05, one is just less than 0.05. How do I feel about that? My view would be that these two slopes are very similar, as I would expect, because there is really a relatively small acute effect. Whether you include the first 6 weeks or not shouldn't make a great difference to the slopes. And as the two arms have very similar acute effects, the treatment benefit should be pretty much the same in the two. So the two p-values being above and below 0.005, I think says that the chance of the slope data arising by the play of chance is about 5%. Having said that, taking the data on this slide together in totality, the question is, does this treatment effect arise from the play of chance? And I would say it does not. And I'll go on to give you some more evidence for that view. We've done subgroup analysis of slope by simple baseline criteria. Here, as an example, is looking at the slope observed benefits by baseline GFR above and below 60. And you can see that the point estimates of these remain remarkably similar for both the chronic and total slopes. Error bars widen, of course, as these subgroups have fewer patients, but there's no suggestion here of a predominance of benefit in one or other strata. Likewise, the bottom two rows show the slope data for baseline proteinuria above or below 1.75 gram per day, and likewise, the benefit does not appear to be different when comparing those two groups. So consistent treatment effect across disease severity. A number of sensitivity analyses have been performed with respect to the GFR slope. Here is, I think, the two most important examples of that. The analysis for the data I've shown you was based on treatment data, and when patients stopped treatment, any further results were not included. The ITT analysis in the top row here shows what would some consider to be the gold standard, where all data was considered whether or not patients had stopped treatment. And you can see here that the benefits of sparsentan appear to be very similar, they're slightly higher, and now the confidence intervals do not cross 0, suggesting significance would be obtained if tested. Similarly, a rescue analysis was performed where data after the initiation of rescue immunosuppressive therapy was censored, so not counted in the analysis. And you can see again that the two treatment effects remain very similar, slightly higher for chronic slope, but now neither of the error bars cross the 0 line. So we think it's a robust result, the benefit in slope, and interestingly, that the intentions to treat analysis and the rescue analysis show somewhat improved changes in GFR with sparsentan. How does this matter? You might think that a benefit of 1.1 mil per minute per 1.73 meters squared per year doesn't sound a very big number, but the accrual of that benefit over time means that the predicted date of dialysis requirement is significantly impacted by the treatment. And this assumes linearity of eGFR decline, which there is good evidence from registry data to support. And you can see here in the green line is the predicted trajectory of renal function in the sparsentan arm. And in the grey line in the middle, the predicted trajectory for the irbesartan arm, increasing the time to need for dialysis by 5 years. We've also put a blue arm there, which is where we believe RAS standard of care is currently achieving a treatment as a slope. And this has been obtained by looking at 5 different clinical studies with similar entry criteria to PROTECT. And the slope is minus 5.3 with a loss of renal function to dialysis within 8.9 years. These plots emphasize the strength of the PROTECT study in that by titrating irbesartan to a maximum tolerated dose in the context of a clinical trial protocol, we believe we've achieved, even with the control arm, the active control arm, better outcomes than is currently achieved by standard of care, and that our active control arm represents a very high bar for sparsentan to be judged against. Further data here, Kaplan-Meier, of a composite kidney failure endpoint, confirmed 40% reduction in eGFR, end-stage kidney disease or death. There was only one death, so this is really about renal function. And you can see that in the green, the sparsentan hazard ratio for this event is clearly lower than that for irbesartan. This difference appears beyond 6 months and is continued throughout the 2-year period. The hazard ratio for that is 0.68. And if you use a 50% reduction in eGFR, data not shown, but the hazard ratio for that is 0.55, in each case favoring sparsentan. These individual hazard ratios estimates and their confidence intervals do cross unity, and therefore they are not individually significant, but they represent a reassurance that the treatment effect we've seen for GFR is likely to be correct. The initiation of immunosuppressive therapy at the judgment of investigators is shown here over time. And you can see that in immunosuppressive therapy was initiated much more frequently in patients in the irbesartan arm than the sparsentan arm. Sparsentan is the lower line in green, by a factor of about 3. So sparsentan treated patients had less exposure to immunosuppression, almost entirely steroids, in the study. Safety is summarized here. Sparsentan appeared to be well tolerated. Comparison in the red boxes of any treatment emergent adverse events or serious adverse events show comparability. And overall, the safety profile of sparsentan looks to be good. I should just add here, because I'm showing you some busy slides, that this talk with the slides will all be placed on the web by Glomcom over the next few days. So you don't need to sort of try to remember all the numbers here. We're going to be showing you these, and they are also published in the relevant publication by Rovin et al. Importantly, transaminase elevations were not distributed towards the sparsentan arm. there were actually 5 versus 7 in the irbesartan arm, so we did not see a signal of transaminase elevation. Peripheral edema was similar in both groups, with no increases in body weight. And the transaminase elevations were not associated with raised bilirubin, and there were no cases of drug-induced liver injury with sparsentan. So here are some conclusions from the PROTECT study. I'm not going to read them all out, but the primary endpoint was met. We believe the clinical benefit was confirmed by the GFR chronic slope, and this in turn was confirmed by a sensitivity analysis looking at the intention to treat analysis. And we believe it suggests a clinically meaningful difference between sparsentan and irbesartan in the eGFR-based endpoints, including a composite kidney failure endpoint. Notably, the patients treated with sparsentan had one of the slowest annual rates of kidney function decline seen in IgA trials. And linked to that, the irbesartan arm also did better than historical and recent clinical trial control arms. So the 1.1 ml per minute improvement in the 2 arms actually represents a 30% reduction in the rate of progression of kidney function loss, which we think is a very significant potential benefit to patients. All the efficacy endpoints at the top line favored sparsentan, and tolerability and safety appeared to be good. So we'll get back to discussing anything that you want about sparsentan in the PROTECT study, but I'm going to now briefly cover a couple of other areas of study which are ongoing, but where we have been able to present some interim data. And I'll talk first about the Spartan study. This is a study of sparsentan in patients with incident IgA nephropathy. So the study is an independent research project being run by the University of Leicester, led by John Barrett and Shikha, with collaboration from other UK centres, including notably Cambridge and Edinburgh. The study is a single-arm study in which sparsentan is provided as the initial therapy for patients diagnosed with IgA nephropathy. And you can see on the left-hand side here the eligibility. Adults, biopsy-proven IgA within 6 months, proteinuria above 0.5 gram per day, eGFR above 30, and importantly, they have not been experiencing treatment with RAS inhibition. The key endpoints of the study, clinically, safety, proteinuria change from baseline, complete remission of proteinuria, and the eGFR and blood pressure from baseline. But in addition to those events, as you can see in the schedule in the main Gantt type chart here, the study is going to include some significant medical deep dive into what might be happening in these individuals. There will be measured GFR measurements made in the early part of the study, up to week 24. There will be total body water measurements by bioimpedance. There'll be renal and cardiac MRI, focusing on fibrosis and function. And there will be a kidney biopsy, a second biopsy at 24 weeks, which can then be compared with baseline patient by patient. These are some of the baseline characteristics, typically male, age about 36, predominantly white race. The systolic and diastolic blood pressure, 125/78, so moderately controlled, but not at target for these patients. eGFR mean of 70, and the median UPCR 1.3. I'll show you some interim results from the study. Here is the proteinuria at treatment, and to remind you, this is a monotherapy. These patients are not receiving any further or rescue treatments. Proteinuria reduces significantly over the first 4 to 6 weeks by about 60%, And over the subsequent period, up to 36 weeks, the proteinuria reduction continues to increase. And at 36 weeks, the change from baseline, the mean change from baseline is 81%. These are the individual proteinuria plots. And I think there are two important things about this. One is that the efficacy appears to be clear in those both above and below 2 grams per day of proteinuria. And you can see those patients who started at or above 2 showing strong reductions in proteinuria. And secondly, that the significant number of these patients, in fact 2/3 of them, at some point do get a complete remission, which, given where they started, is a significant therapeutic achievement. So it's a small study. The data is promising. We're very excited to be working with Leicester to understand more about these patients, and particularly to have full data on the second biopsy that will give us a lot more mechanistic information. These are the eGFR changes in Spartan. The acute effect of starting the drug is perhaps remarkably not very great, maybe around 2 mils per minute over the first 6 weeks. And over the subsequent time, up to 36 weeks, eGFR broadly stable. Wide error bars, very small number of patients. This is not the basis for an efficacy statement, but it does look that there is safety in terms of GFR profile over this period. These are the blood pressure reductions, and you can see that on starting Sparsentan, there's a reduction in blood pressure. It enters a controlled phase and then appears to be stable over time in the first 36 weeks of this study. One patient did have to withdraw because of hypotension and appeared very sensitive to Sparsentan, and that's something which needs to be borne in mind in the drug's properties. These are the initial data from bioimpedance. On the bottom here, total body water, and you can see that over the initial period of the study, up to 24 weeks, change from baseline of total body water is negative. These patients do not appear to have any issue with any increasing edema, perhaps even the opposite. They seem to be losing body water, and their weight remains stable. So this looks like a positive tolerability and safety profile. So these are the safety bullet points. Well tolerated over 36 weeks. I mentioned the patient discontinued due to hypotension. There have been 3 SAE, but none are considered related to treatment. So here's a study summary of the interim data from the Spartan study. This is first-line treatment, newly diagnosed patients with IgA, and the agent appears as monotherapy to be effective at reducing proteinuria and controlling blood pressure. Effects on proteinuria were substantial, and Sparsentan treatment was generally well tolerated. The rapid and sustained reduction in proteinuria and other findings of this study are comparable, as I think you can see from what I've shown you, to those from the phase 3 PROTECT study. And the further planned analyses are really very interesting as we look to understand in more detail the biology of Sparsentan in human clinical use. So finally, just for 2 or 3 minutes, then I'll give you guys a chance to speak. I'll cover 3 studies which are ongoing, looking at Sparsentan in combination with SGLT2 inhibitors. The PROTECT study did not allow the use of SGLT2 inhibitors in the double-blind period. It started well before that became a realistic treatment choice, and we felt it was not possible to change it as we went along. However, the open-label part of the PROTECT study does allow SGLT2 inhibitor use, and we're going to utilize that in a range of ways to study the safety and efficacy of that combination. First of all, we have analyzed those patients in the open label who, under a physician choice, were started on SGLT2 inhibitors. And apologies for the busy slide, but the bottom line here is that in the open label of PROTECT so far, 39 patients were initiated on SGLT2 inhibitors by physician choice. They appear to be tolerating the combination well in terms of blood pressure, body weight, stability, and the urine protein creatinine ratio tends to fall over the period of observation. I'm sorry that the axis on this graph in the middle appears to have gone a bit awry, but the change is actually from a median of 1.8 to a median of 1.2 over this 48-week period. So it looks as if the effect of the SGLT2 inhibitor initiation is about a 30% reduction in proteinuria, and that would be, I think, consistent with an additive effect of the combination on proteinuria, given what we know from the SGLT2 inhibitor studies themselves of about a 25 to 30% reduction in proteinuria. So this is ongoing, but at the moment we feel it's demonstrating tolerability, no safety issues have emerged, and the provisional evidence that the combination looks to have additive efficacy in terms of proteinuria. In order to make that evidence base stronger, there are two studies ongoing. On the top here, we have a PROTECT open label sub-study. And in this case, patients in PROTECT open label on Sparsentan are being randomized to receive SGLT2 inhibitor or not for a period of 12 weeks. And then at the end of that period, have the option of continuing or not their SGLT2 inhibitor. So we get two bites at the cherry, in a sense, for looking at the effect of starting SGLT2 inhibitors in comparison to not doing so in this study. We don't have data from this yet, but we are interested to analyze this likely later in this year, early next year, to demonstrate whether we have tolerability, safety, and what the efficacy of the combination looks like. And then in a complementary way to that, the Spartacus study is an exploratory open label design where patients who are stable on SGLT2 inhibitor with IgA nephropathy in a prevalent phase are given in a single arm design, Sparsentan, and this with the usual titration up from 200 to 400 will allow a assessment of the efficacy of adding Sparsentan to patients on established SGLT2 inhibitors, as well as looking at, again, safety and tolerability. So lots to come, and we're looking to build up the evidence that the combination is something that could be considered when Sparsentan is used. So my final slide. We have, from the studies I've shown you, consistent evidence of efficacy for Sparsentan, substantial reductions in proteinuria and protection of renal function. Tolerability and safety appear to be consistent and manageable. The efficacy of Sparsentan appears to be present in both incident and prevalent patients, and in effect, the efficacy independent of baseline levels of proteinuria and eGFR. And in the data we have so far, combination of Sparsentan with SGLT2 inhibitors does appear well tolerated, and preliminary data suggests that the antiproteinuric effects are additive. So we think, taken as a whole, Sparsentan has growing evidence base that it could be considered, perhaps in the future, a foundational treatment in which patients with IgA receive something which is going to be a basic foundation treatment from which other treatments can be added as needed, but that the switch of RAS inhibition to Sparsentan provides a very clear benefit in terms of renal function preservation over time. I shall stop there, and I look forward to taking part in discussion with my colleagues Kirk and Shikha.
Excellent, wonderful. Thank you very much, Bruce, for a thorough and in-depth review of the clinical data, and also both on the PROTECT study and also the other studies currently ongoing. We have few questions here from the audience. We'll bring that forward. Looking forward to the input by Dr. Campbell and Wadhwani. Maybe starting off with one question, and that is the on a clinical from a clinical side, one thing we are very interested is the renal protective effect, GFR decline. And we have here now the issue of around the chronic slope and the total slope and both the clinical, the statistical significance, the clinical significance, and also the issue around around the approval and long-term outcomes. Can you help us a little bit understand the difference between the chronic slope and the total slope, and which one should be considered and is being considered with regard of efficacy outcome?
I certainly try to give you a shot at that. First of all, let's consider chronic slope. This is the slope where the data in the first 6 weeks is not included effectively. Data from 6 weeks, 110 weeks is included. And the idea is that chronic slope contains information about the trajectory of kidney function over time on drug and does not include any acute effect of the drug. Whereas, and this is important when you consider drugs that might have acute effects on GFR that may be a one-off. They may not continue, they may not change over time, but there's an acute effect, but they're not really predictive of the long-term effect. Total slope is saying, let's not worry about acute effects, let's not worry about mode of action, let's just take the total effect of the drug over 2 years. Perfectly rational thing to do. The issue arises when there is an acute effect. So to give you an example, if there's a negative effect on GFR in the first 6 weeks, then the total slope will give you an underestimate of the trajectory benefit on kidney function. Because if you try to take the total slope and predict the future, you're effectively assuming that the acute effect will be repeated every 2 years, which is clearly not going to be the case. So the total slope has the problem that its ability to predict the projection of kidney function over time is limited by including the acute effect in the slope calculation. The chronic slope improves, logically, I believe, the ability to predict the trajectory for the future. Conversely, if the acute effect is positive, then the total slope will overestimate the likely trajectory treatment benefit of the drug, because again, if you just multiply total slope with longer times, you're essentially assuming the acute effect will be repeated every 2 years, unlikely to be the case. And the two examples of that are bardoxolone and steroids, where acute effects tend to be positive. My view is that if you really want to understand the future trajectory, you're better off using chronic slope. Now, it turns out in the PROTECT study that the acute effects were really quite small and didn't differ between the arms. And it follows arithmetically that the chronic and total slopes are actually very similar, and the differences between them are almost negligible, 1.0 versus 1.1. So in the PROTECT study interpretation, there is no competition between the chronic and total slope. They're both telling us that the slope is improved. Now, the question of whether the p-value is above or below 0.5 is a different discussion, I think. But in the PROTECT study, both of the slopes are providing evidence for a treatment benefit, and they are close to each other because the two arms of the study have a very similar acute effect, and which in turn is not that great. I better stop there because I should let someone else speak.
Great. Thank you very much. With regard of this acute effect and then the long-term effects, I have two questions. Maybe one for Dr. Wadhwani first, and then the second one for Dr. Campbell. So the acute effect, Dr. Wadhwani, is, as Dr. Hendry mentioned, the acute effect is minimal. It's similar to between the control arm and Sparsentan, but we see kind of a GFR drop, and is that an hemodynamic effect? Do we see that in other renal protective clinical trials as well? And what does that effect potentially do? And how to worry about or how to deal with that GFR drop in the clinical setting when we care for the patients?
Well, thanks so much for having me, Ali. And you can hear me okay?
Yes.
Great. So, I think this question about that acute drop in GFR is not something new to us as nephrologists. We're used to this with RAS inhibitors. We see this with SGLT2 inhibitors. And I think we really need, we're used to, or we're working on educating our patients about this, as well as other primary care providers, that this is not something to be alarmed about. This is an expected change, and it should not lead to discontinuation of any of those three drug classes. Now, when it comes to the fact that, is this just a hemodynamic effect? I think there's two things I would look at. First of all, if you look at the blood pressure differences in the PROTECT trial between irbesartan and sparsentan, you don't actually see a significant change between the two groups in terms of blood pressure. So I think that's one thing to consider. And then when you look at the proteinuria difference, there's a significant drop in proteinuria even in the first 4 weeks in the patients on sparsentan compared to irbesartan. So if this was just a hemodynamic effect, I don't think you would expect to see that. I think, as Bruce nicely showed, there are a lot of other parts of the glomerulus and the nephron that endothelin is involved in. And when you're looking at some of that preclinical data, you do see that there is an effect on podocyte number, glycocalyx, et cetera. And so I think there is accumulating evidence that this is more than just hemodynamics.
Great, thank you very much. And that helps me actually go to the next question that I had for Dr. Campbell. Dr. Campbell, you're the foremost, podocyte biology researcher and basically spent a lifetime career on understanding the pathophysiology of the glomerulus and the podocyte. So what is that? And Dr. Hendry, can you please put the slide with the Kaplan-Meier curve on the complete proteinuria remission out there? If you can share that. So with regard of the reduction both in proteinuria and the renal, the reduction in proteinuria that is substantially more in the sparsentan group, irrespective of the blood pressure hemodynamic effect that we see, because both groups, as Dr. Wadhwani mentioned, have equal proteinuria, equal blood pressure effect. How do we understand that? What is the mechanism of sparsentan on the podocyte or on the glomerulus beside the hemodynamic, or is there a different hemodynamic mechanism? Number one, that, and number two, is it seems that, and I meant your other slide, which one was that? That your time to event analysis for the complete remission, proteinuria remission, Bruce. I think it's one slide after more, next. Yes, is that? No,
yeah,
that's the composite.
This is the composite endpoint. I didn't show
this
you
one.
Kaplan-Meier.
There you go.
This is to initiation of immunosuppressive therapy.
No,
I didn't show you the Kaplan-Meier. We do have that.
Okay.
And if you look at the Lancet papers from 2023, you will see some data on the time to proteinuria remission. But essentially, proteinuria remission is occurring, the difference is occurring early and is sustained and accumulates during the study. And the sparsentan arm is clearly superior. Both remission below 0.3 and remission below 1.0 grams.
I think it was probably, I think it was in the supplements of the exactly. Published study. So it's in the supplements of the study where you can see basically even after the first year, patient in the sparsentan arm, even after the first year, continue to achieve complete remission. So basically suggesting that there must be something else going on. So what is going on in that glomerulus? So do we know it?
Yeah, no, thanks, Ali. Great question and certainly great presentation by Bruce and great comments from Dr. Wadhwani as well. I mean, I think as you alluded to earlier on, endothelin 1 as well as angiotensin 2, right, are kind of multifactorial promoters of glomerular and tubulointerstitial injury. So it's not specific to podocyte protection, we believe, for this dual acting antagonist. We must remember there are other endothelin receptor antagonists in development in the kidney space, but this is an angiotensin receptor blocker, endothelin receptor antagonist combination. So you are getting protective benefits on the vasculature, podocytes, the endothelium, tubulointerstitial compartment. It's beyond hemodynamic benefit, one would believe, right, in this case, based on the mechanism of action of the drug. And it's not surprising then that you see the sustained benefit, right, over time with prolonged use. And again, I guess there is certainly remodeling happening at the cellular level, right? We know that podocyte depletion may be irreversible, but if you identify patients early and you can protect podocytes from ongoing injury, you can potentially, by reducing proteinuria, albuminuria as a marker of glomerular injury, slow the progression of disease over time, right? And this is where it gets to the GFR slope protective benefit as a functional readout, if you will, of the overall, intact nature of the glomerular filtration barrier.
Ali, would you like me to add to that?
Please. Yeah, I love an open discussion.
So first of all, I'm pleased that we seem to have a consensus that the benefit above irbesartan is not hemodynamic, and that's an important point that the data strongly supports. Sparsentan has a hemodynamic effect. Importantly, it controls blood pressure, for example, largely probably because of the AT1 antagonist properties. But its hemodynamic effect is very similar to irbesartan. So its additional and superior benefit in the study is not hemodynamic. So what does move the needle? I mean, Shikha and Kirk have already mentioned a couple of these things. Let me give you an idea where I think we are. In the first few weeks of the study, 4 or 6 weeks, proteinuria is clearly reduced, 40% plus. What could do that? Well, 2 things come to mind. One is a direct podocyte protection, where podocyte cytoskeleton can have improved structure and the slit diaphragms can actually be more integrated and functional. And the second is that the protection of the glycocalyx can definitely occur within that time scale. Both of those 2 effects are dynamic and could occur within weeks in the context of initiating sparsentan. Then, as you mentioned, Ali, there seems to be even a further accumulation of benefit over the first year and indeed in through the second year. What other factors could be playing there? Well, I'll give you 3 of them. One is sparsentan is anti-inflammatory, both at the cellular and the cytokine level, and it also reduces the transcription of key complement genes. So there's a whole inflammatory component, I believe, to the effects of sparsentan. And again, preclinical and cellular data is supportive of that. The next effect is that sparsentan is anti-proliferative. So it does reduce proliferation of mesangial cells in vitro work. And it looks as if that effect occurs in vivo in preclinical data. And that will be reducing one of the drivers of glomerular damage, because we know that mesangial cell proliferation is a hallmark of the progressive disease IgA nephropathy. And then finally, as Kirk has already mentioned as well, sparsentan is anti-inflammatory. Sparsentan is anti-fibrotic. And we know that fibrosis in the glomerular context and also in the tubulointerstitial compartment is likely to be a part of the driver for loss of kidney function. So I think the reason for the accumulation of benefit is over time a combination of those 5 things: podocyte protection, glycocalyx preservation, anti-inflammatory, anti-proliferative, anti-fibrotic. Now, I can't prove that that's the case or which is the most important, but I think it all is consistent with the data I've shown, and the Spartan results over time should give us a lot more information about where I'm right and where I've misjudged that. But I think it's a very, very realistic working hypothesis as to why we get the accumulation of benefit over such a prolonged period of time.
Great, thank you. So in the PROTECT study, there was no subgroup who was biopsied and follow-up, or where we have follow-up biopsies?
No, there is no further biopsy.
But Spartan hopefully will answer that question. So do we have any subgroup, like, in the clinical setting, we look at hematuria often as a kind of a surrogate for disease activity, often associated with inflammation. Do we have any data on hematuria?
So we have the baseline data. We do not have follow-up hematuria data. We are looking into that, but as a multi-center global study, the fidelity of hematuria data is, to our mind, a little bit questionable. So we're reluctant to base too much optimism, if you like, on the hematuria on drug in the study.
Okay, understood. So basically the Sparsentan group had less use of rescue steroid therapy. So and and how do you see that, Dr. Wadhwani and Campbell, in your clinical practice with regard of is that a steroid sparing, non-immunosuppressive medication? Which patients do you think about using it? Do you use it in conjunction with steroids or other immunosuppressive therapeutics? How do you put it in the landscape of the other drugs that have been approved or are on the horizon?
Yeah, so great question, Ali. I think our understanding of the therapeutic landscape of IgA nephropathy is really evolving. And with new data that comes out every few months, you need to kind of stop and reorganize how to interpret the data, how that adds to what we already have in our armamentarium. And I think for me, when I look at the data for Sparsentan, and I have that as an option now, at least in the United States, compared to, say, a RAS inhibitor as my background therapy, the data sort of speaks for itself. There is greater proteinuria reduction, there's greater GFR preservation, it's well tolerated, and you can actually get it fairly easily. I actually was able to get it for a patient within like a month of biopsy, and I did not have to wait 3 months of traditional RAS inhibition before starting Sparsentan, which to me, why are we waiting 3 months when we have something that we can use now and is superior than, a RAS inhibitor alone? Now, when it comes to, individualizing therapy for IgA nephropathy, I think we have to remember this is a heterogeneous disease. Every patient's going to look a little bit different. Where they're at the time of their presentation to you is going to be different. So the time of biopsy, we have to remember, is not the time of disease onset. There's a huge latency period, in part because patients aren't getting urinalyses regularly, at least not in the United States. Like if you go out to, let's say, Japan, they have screening tests and they're getting urine by like the age of 5 or 8, and they're identifying microscopic hematuria. But here, patients often don't come to our attention until someone notices that the creatinine's rising and then thinks to check the urine, or they go in for an insurance test and they happen to have an abnormal urine sample and then get a workup. So I think that when you think about the fact that the majority of patients by the time of diagnosis of IgA nephropathy are already at CKD stage 3 or worse, at least in the United States, they have already had a significant amount of not only glomerular, but tubulointerstitial injury and fibrosis at the time of their diagnosis. And so I think you need to be thinking about the kidney injury aspect, so that hit 4. And then if there is significant endothelial proliferation, when you look at your MSC score, if they have an M1, E1, C1, you also need to be thinking about that. How am I going to target that? And I think these drugs can be used in combination. There's no reason why you couldn't use Sparsentan alongside, say, a corticosteroid. Maybe what you may want to do is start the Sparsentan as your background therapy, get the biopsy if you think there's more that's needed, do that next step, whether that's going to be, corticosteroids, whether that's going to be budesonide, whether that's going to be a clinical trial. We have multiple trials now looking at B-cell therapies, complement inhibitors, et cetera. I think you can tailor that to each individual patient, but I think this is a good background therapy to prevent long-term kidney injury and progression.
Thank you. And for the clinical purposes of practice, there's a question by Dr. Lau here. How low of a GFR do you feel comfortable starting a patient on Sparsentan?
I'll give you a quick answer to that. The study was designed to take GFR above 30 at the starting point. In practice, because of differences between the screening and the baseline, some patients between 25 and 30 were also recruited. No safety issues have emerged in that group. As we look at the open label extension, we're going to have data on starting the drug below 30. And so we're looking to bring that forward whenever we have a chance to do the analysis. Patients on the study have, in some cases, developed GFR stage CKD4. We have not had any safety issues with that. And the label in the United States doesn't exclude use of the drug in CKD4. So I think it's up to individual physician decision where they think it's reasonable to start the drug. My own thought at the moment is that certainly above 25 is a reasonable place, but I'm interested to see what Shikha and Kirk think.
Yeah, I'm just gonna, really agree with what Bruce and Shikha just mentioned. I think we have the benefit now of several agents that have an efficacy signal that appear to be well tolerated. That's great for patients and great for our clinical practice. I think going forward, we can potentially be guided by a more proximal kidney biopsy result, as Shikha alluded to, right? And studies like Spartan and kind of going back and taking a deeper look at the biopsy vintage, how proximal to the study enrollment, do you have biopsy data available? Should we consider repeat biopsies in patients who were diagnosed too long ago? Because evidence of active inflammation, right, we know portends a higher risk of progression, right, from the, again, risk prediction tools and so on, and certainly our own clinical experience. So we may want to be more aggressive, right, earlier on when patients have evidence, right, of active inflammation on the biopsy in combination with other clinical readouts, active urine sediment, high-level proteinuria, GFR that's already declining. And, we may not want to try one agent, then add another and wait 6 months, then wait, we may want to really use combination therapy up front. Certainly something or a combination of agents that would reduce the proteinuria level by up to 50% off the bat, certainly would appear more attractive than a single agent that would do so at about 30% or so. And certainly thinking about, as Shikha mentioned, complement B-cell targeting agents in development are going to be also useful to use as a part of a multi-target approach, especially for those at higher risk of progression.
Can I just piggyback off of something Kirk said? I think that big picture in 2024, what we're realizing is, I hope that IgA is not as benign as we thought it was. And I think a lot of the data that's come out from, Dr. Barrett in the UK radar registry, as well as the data that's been, shown here in the United States and around the world, is that getting proteinuria down to just less than 1 gram per day should not be our target. And I think the updated KDIGO guidelines for IgA nephropathy that'll hopefully be out soon are going to reflect this. When you look at the UK radar registry data, for example, something like 20% of patients, even if they got their proteinuria down to less than 0.5 gram per day, still had 20% of them developed end-stage kidney disease during their lifetime. And then if you look at, there's a night heat map in that picture paper, and it shows the degree of GFR decline you need to achieve in order to prevent ESKD in your lifetime. And you really need to get that GFR slope to less than 1 ml per minute per year, depending on the age. And what I think we're realizing is we're not going to be able to achieve that with any one agent, as Kirk said. So it's going to take a multi-targeted approach. I think that we're really, something that I've been thinking about in nephrology, but specifically within IgA nephropathy, is sort of goal-directed therapy. Like, what should our goal-directed IgA nephropathy therapy be? And it's probably not going to be just one agent. When we have learned so much about the pathophysiology of this disease, and there's multiple hits, why are we not targeting more than one of these hits? Again, individualizing it based on biopsy, clinical characteristics, patient preference, etc.
Great point. Multi-targeted therapy will be a theme for glomerular diseases all in general, as we have also seen it in lupus and ANCA vasculitis. To that theme, so one question when I say that came out from the participants was, is there an indication or are there clinical trials of Sparsentan in other glomerular disorders currently going?
So we have a ongoing study in FSGS, the duplex study, which has reported the 2-year double-blind period data in the New England Journal of Medicine in November last year. The duplex study, I'll give you two key facts about it, is that the antiproteinuric effect of sparsentan, again against an active control irbesartan, was again very clear. So there was a 50% reduction from baseline in the sparsentan treatment arm. And the second thing about the duplex study, which I think is noteworthy, is that the safety tolerability of sparsentan was pretty much the same as irbesartan and confirms those data from PROTECT. However, in the duplex study, the confidence intervals for eGFR slope were twice as wide as we see in PROTECT. They were plus or minus 2 mil per minute. And the treatment effect on eGFR slope in the duplex study did not achieve statistical significance. So the duplex study is now being looked at in some detail to see what it tells us about the treatment benefit of sparsentan. And at the moment, we are in the process of that analysis. There is a process that has been kicked off recently, partly with the duplex result in mind, led by NephCure, to look at what should be the endpoints for studies in FSGS, because I think the duplex study has given us pause for thought about that question. And my own view is that we should be looking in detail about proteinuria efficacy, coupled with some form of GFR signal, perhaps in the way that we do in lupus. But that is something being worked on very actively by NephCure and their collaborators. And we at Travere are very keen to be helpful in collaborating with that as it goes forwards. I'm interested because of Kirk's interest in FSGS to see what he thinks about this, so I shall stop there.
Yeah, no, and yeah, both Shikha and I are part of that initiative as well, the Parasol supported by NephCure, because I think that the regulatory landscape for IgA nephropathy drug development benefited tremendously from developing that path to accelerated approval by accepting proteinuria reduction as a validated surrogate marker, right, of efficacy. And again, we've seen the major benefit for patients that's resulted from this. So we really need to define, again, those endpoints more carefully for FSGS. And, again, a 50% reduction in proteinuria, is very impressive, right? And one wonders, right, whether or not, you can project out for what may be lifelong therapy, for individuals, if you would see a significant benefit to eGFR, slope over an extended period of time. But of course, you can't responsibly design a trial to be ongoing for 5 to 10 years. So we need certainly to have these validated surrogate endpoints that are achievable in a feasible manner in the context, right, of a clinical trial. Our patients really deserve that. And again, just so everybody is on the same page, we have no FDA-approved therapy for FSGS, which is quite sad.
Thank you. Thank you very much, Dr. Campbell. And without distracting too much from the IgA topic, was the issue with duplex an issue of the endpoint, or was it an issue of the heterogeneity of the disease and the difficulty of dissecting FSGS into concrete categories with all not being the same?
I think that is a very good question. The duplex study did include both genetic and primary FSGS, but I think the problem is more overarching. The FSGS clinical course is more episodic in terms of remission and relapses. And so the individual patient data suggests eGFR varies a great deal more over time. And so an eGFR slope analysis becomes much more problematic. So I think there's a range of issues there, including heterogeneity and the nature of disease, that make eGFR slope a much more imprecise endpoint for the FSGS cohort.
Okay. And one more question to that topic. So was there a maximum difference in maximum tolerated dose of sparsentan in PROTECT versus duplex?
So duplex used double the dose. In other words, it started at 400, titrated to 800. That was because we felt with a protein-bound agent, it might need really the most we could give to get it to the target cells in the presence of hypoalbuminuria. In fact, the pharmacokinetics suggest that the exposure to the drug does not go up proportionally. But importantly, the duplex data showed no additional adverse events or additional safety or tolerability issues at the higher dose.
Okay. So, okay, great. Thank you. So I have one more question before then leaving it to Dr. Campbell, Wadhwani, and Hendry for any concluding comments and remarks. Is, you know, in the PROTECT study, only 60% entering the study were prior on maximum tolerated ACE/ARB therapy. Roughly 50% only were on statins. And even the irbesartan group showed a reduction in blood pressure, proteinuria when put on maximum tolerated therapy. So how much are we behind with actually implementing standard of care in patients with IgA nephropathy, and what do we need to do better?
Just want to briefly just correct that slightly. The percentage you quote is the percentage on the maximum labeled dose. In fact, they were almost all on what was believed to be maximum tolerated dose when they began the study. That was a criteria for entry. 6% were on maximum labeled dose. But clearly, starting the study and titrating up irbesartan did create an improved outcome compared to what looks like standard of care. And I think it shows us that the practical achievement of standard of care, real maximum tolerated in real life practice, is perhaps not as good as we would like it to be. But interested in Shikha and Kirk's thoughts on that.
Yeah, I would agree. So I think I joke that what PROTECT taught me is irbesartan's a really good ARB, but I think that's because in a trial, you have very good compliance. These patients are being followed very closely. You're counting pills. And this study was very unique in that it had an active control arm. And so these patients who started off at what was thought to be 60%, 60% of them actually were on the maximal label dose. I'm saying that correctly, Bruce, at entry into study. But then once they were in the study, 95% or so were able to tolerate the full 300 milligrams of irbesartan or the 400 milligrams of sparsentan. And so what I think that tells us is exactly what you said, Ali. We're not really getting patients to the maximum tolerated dose in our own clinical practice. And I think that's really important as you're considering other trials in this IgA nephropathy space as well, because they're being compared to placebo on a background of standard of care RAS inhibition, which is maximum tolerated. However, this study showed us that maximum tolerated that we think as nephrologists is not really often what patients can handle. So I think when you're looking at comparing things, that's something that we all need to keep in mind. This was a very, very rigorous study that did have an active control arm. And that's why you see that additional 15% drop in proteinuria at the first 36 weeks, which you wouldn't otherwise expect if these patients had been on, you know, a really good dose of RAS inhibition for the last 3 months before entering the study.
And yeah, it's definitely not a new phenomenon. We knew from the Stop Icahn study, really more than 30% of patients didn't qualify for the study once they're optimized on RAS blockade, right? So we clearly have work to do in optimizing standard of care therapy that's foundational.
Thank you very much. Great. I mean, we are 13 minutes past time, and I still have a list of questions, and people also bring up questions. Perhaps just leaving it to any final remarks, concluding remarks, Dr. Campbell.
Yeah, no, I'd say again, this is good for patients that we have options that are safe and efficacious, and we now are challenged, right, to define which patients will benefit the most from which therapy and in which combination. But that's not the worst problem to have. It's much better that we be here than where we were 5 to 10 years ago.
Excellent. Great. Well, thank you so much. Thank you, Dr. Hendry, for your presentation. Thank you so much, Dr. Wadhwani and Campbell, for being the panelists, and for all the participants here engaging and joining us today. Have a
Thank you
wonderful
to you,
rest of your
Ali,
day.
and been a great pleasure. And thanks to all the active participation. Thank you very much.
Thank you.
Yeah.